Hello everyone. Today we'll be discussing about the CADD or drug design, that is revolutionizing medicine discovery. CADD means computer-aided drug design, that is transforming how we discover and develop life-saving medication. Actually, in the previous time, whenever the newer synthesis process was there, the medicine discovery process was traditional. So in that time, traditional drug discovery are notoriously slow and expensive and often taking 10 to 15 years and costing billions dollars to bring a single drug to market. And failure rates remain high throughout the development pipeline. Because in this previous time, the synthesis process was skimmed out, then the synthesis process was carried out in the lab, then different chemicals, different agents you have to follow. And next, if there is any problem in the procedure, then the drug will not be or synthesized. And after the synthesis, there were lots of formulation parameters trial, then the experiment study. So there were lots of chances where the drug synthesis was maybe for. So that's why the need for the change has came up with the recent years. So the pharmaceutical industry urgently needs faster, more efficient methods to combat emerging disease and addresses unmet medical needs. Time is critical when lives are at a stake. So in this modern phases, we have seen that the diseases are coming in a very faster way, and there are different diseases and very tough diseases which cannot be treated with single drugs, and that's why we need more drugs which are more potent and more precise to the diseases. Nowadays, it has been found that the different microbes which are changing their structure very fast. So that's why antimicrobial resistance is occurring in patients. So the we know that the bacteria has a very tendency that they can change their genes or the cell wall very fast. But to kill that bacteria or change the structure or to inhibit the growth, the process of synthesis or discovery of the drug is very tough. So it takes 10 to 15 years minimum. So that's why the drug discovery process is used by the computer-aided drug designing. So that leverages advanced computational methods to accelerate the drug discovery and development. So that is facilitating accelerating discovery, rapidly identifies and develops promising drug candidates using virtual screening and molecular modeling. Integrated approach that combines molecular modeling, computational chemistry, and rational drug principles for optimal results. So in this process, suppose there were some molecules like sulfonamides or quinolone that were previously used for the malaria, but nowadays it has been found that the sulfonamides or malaria quinolone molecule is not treating the effective the malaria or the different bacterial infection. That's why scientists are thinking that if there is some modification or substitutional changes in that structure, that can give the more potent drug or more efficacious drug. So that's why they are not going for the lab synthesis. So they have developed some software through which if the structure has been designed and the modification or substitution in different places of the drug is done through the computer drug design, and different parameters like the lipophilicity, hydrophilicity, partition coefficient, and drug distribution, their toxicity, ADME, everything will be determined through the software. Whether if we just change the substitution for these drugs, then what will be the drugs parameter, whether it will be pharmacologically active or not, whether they will bind to the receptor of the body like this. So if it is predicted very fast, then you can go for the synthesis of drug in the lab. So that will be very faster process. Next, the CAD workflow from heat to lead. In this process, we can see that first of all, we have to go for the virtual screening. So that virtually rapidly screens most molecular libraries, assistant target proteins to identify initial hit compounds with potential therapeutic effect. Here, the protein molecule is important target protein for every compound or every drug that is binding to any receptor or any protein to our body that is having some proteinous nature and proteinous structure. Like for the disease of different infection, the bacteria also having some protein or genetic material. So that is also determined the structure through this computer-aided drug design or some protein data bank from where structure of the protein. Now we have to identify some drug or some molecule should design like quinolone Suppose this is a category. Now we have to bind with that protein, whether the drug can fit with the protein or can inhibit or it can inhibit the different structure of the protein or places of the protein, so that the protein cannot replicate or is destroyed, or the genetic material of DNA RNA is destroyed, and as a result, the disease cannot grow. Now coming to the molecular docking, that examines binding specificity by computationally simulation, how each molecule interact with the target binding sites at atomic level. And now it is also, I have already discussed, like receptors in our body or any protein or any lymphocyte or any lymphocyte or monocyte. So that is also having some genetic material, some yes, or some receptor is there. So whether the drug can bind to that particular molecule or not, then we have to find out. Now ADMET prediction, that is in silico analysis predicts absorption, distribution, metabolism, excretion, and toxicity properties to filter promising candidates. So if the drug is added with that protein, and then their absorption, their distribution, then metabolism and their excretion should be determined through the software, and the toxicity that also be predicted. So if we get the sustainable amount or a reasonable drug, good quantity drug, where all the parameters are good, can synthesize the drug. Now lead optimization. So defines promising lead compound, that is a main compound or the main drug that is to be optimized structures ready for chemical synthesis and experimental testing. So if we have derived the drug synthetically, if we have designed the drug computerized, whether this compound is stable for having pharmacological activity and stability and many more aspects are good, then we can go for the experimental testing and synthesis. Okay. Now, the two pillars of CADD, structure versus result. So there are two methods, that is structure-based drug design and ligand-based drug design. So that relies on the three-dimensional structure of the drug target protein. Target structures are obtained from the protein data bank and predicted using molecular modeling techniques when experimental structure are unavailable. Requires known 3D protein structure and enables rational and structure guided design, more precise targeting. So here we have to target the protein and should get the structure of the protein data bank. That is small in a abbreviation known as a PDB. From that protein structure, you have to draw the protein 3D structure, and you can see the three-dimensional structure. On here enables rational and structure guided design, more precise targeting approach. As a result, if there is a pharmacophore modeling or at the protein data bank, there is a space where the drug can be fit. So if the drug can fit in that protein structure, then we can go for the synthesis of the drug. Now another is ligand-based drug design, that is used when the target's 3D structure is unknown or unavailable. Then instead of focusing on analyzing known active molecules to identify common structural features and pharmacophore patterns. Suppose some disease have been identified like cancer, so where we don't know the structure of the protein, but we know that in case of like spindle cell sarcoma or the breast sarcoma or breast cancer, there is some drugs like suppose we are catching vincristine or flucysteine, like this drug can having activity towards the cancer. So we have to draw the structure, we have to design the structure according to the problem. So here no protein structure required, and based on the known active compounds using QSAR and similarity methods. So that is important. QSAR, that is quantitative structure activity relationship. That is, you have studies the ACR, that is the structure activity relationship, where if there is any modification or changes, there will activity increase or decrease we can found out through the ACR. But through the QSAR, the quantitative analysis, that is the lipophilicity or the quantity will determine the protein partition coefficient, the half-life, everything will be determined through the quantity. And through this QSAR study, we can determine if the drug can act, the drug can be synthesized. Okay. Now, precision targeting, the power of molecular visualization. Advanced computational tools create detailed three-dimensional models that reveal exactly how drug molecules interact with their protein targets at the atomic level, enabling rational drug design. Now, so it is impact. There are very good significance of this method, that is computer drug design: speed, cost, and success. The time saving, it is very time saving, significantly reduces the timeline from initial discovery to clinical trials, compromising years of work into months. Virtual screening can evaluate millions of compounds in days instead of years. Cost effective, lower the R&D expensive by minimizing the costly experimental failures. In silico prediction, that is the in vitro in silico prediction, reduce the number of compounds required, expensive synthesis and biological testing also. They increase success rate and improve the likelihood of the identifying viable drug candidates by filtering out problematic compounds early. And combinatorial and computational prediction catch toxicity issues before synthesis. So these are the very good impact and significance. Now, real world application beyond the lab, we can for the academic research, for identifying and for treating complex diseases and pharmaceutical giants. We can synthesize the pharmaceutical lead molecule by this process, and through this AI integration, we can get the drug synergistically, and synergistic activity of different drugs can be identified. Okay. There are different machine learning algorithms, different softwares are there through which we can identify whether the drug is active or not. The future of drug design integrated intelligence, it's obviously having very good future. They are using AI and machine learning, and personally medicine can be derived also, and it has a novel therapeutics also. Designing tomorrow's cures ends today, and lastly we can say the CAD designing, that is remarkable testament to human ingenuity, combined with the computational power, transforming how we approach medical challenges, revolutionizing drug discovery from a high-risk size rational science with predictable outcomes. So I have to remember this topic, and there are some parameters that you have to remember also. Now I'm going to discuss some points which we have to calculate through the process. Now I'm showing a PDF for you. Here we will be discussing some parameters that you should study during this year. See, this is the drug design chapter that already have been discussed in through the PPT. Now we are some parameters. These are the drug discovery processes, drug discovery 2 to 5 years, preclinical development, then clinical development and regulatory application. That is, there are two major types of approaches to drug ligand drug design and structure-based drug design that I have discussed. Now, just it is an example, pyramid and core using similar structure near both, but some modification is there. Chlorine and the image. So this is the protein structure you can show. This is a three-dimensional protein structure, and this is the ibuprofen, which will be fitting to that COX-2 inhibitor, COX-2. So that is determined through the pi-mol or pyrite software. This is a protein target, and this is a library of compounds which can fit here. That identify compounds that bind challenges. Okay, now different descriptor of properties of features are there. That is some physiochemical properties like electronic factor, steric factor, electricity, hydrogen bonding, shape, charge, polarizability. That you have to determine QSM. Now there are some molecular descriptors like hydrophobic parameter, that is the partition coefficient log P, and substitution constant pi, hydrophobic parameter F dash, distribution coefficient log, solubility parameter log S. Now there are some another electronic parameter Hammett constant, Tufts inductive molecular constant sigma, asterisk, next ionization constant K delta P. Then Tufts steric parameter E is molar volume, van der Waals radius, and molar refractivity that you have to determine this drugs. And energy of EHMO, that is the energy of EHOMO and ELO, energy of highest occupied molecular orbital, and energy of lowest unoccupied. Through this structure, you can get the energy of a drug. There are special descriptors also, Juris descriptor, shadow indices, radiation, direction, flexible molecule, so determined, but for smaller cases we don't. This is the drug. Just 1D, 2D, or 3D, 4D structures is there that we are checking. Now partition coefficient log P, that is very parameter log P, that is the octanol water partition coefficient, is a measure of hydrophilicity and hydrophobicity of the compound. In this physical sciences, a partition coefficient or partition distribution coefficient is a ratio of concentration of a miscible solvent. The ratio is therefore comparison of the solubilities of the solid two liquids. The partition coefficient generally refers to the concentration ratio in unionized species of a compound, whereas the distribution coefficient refers to the concentration ratio of all the species of compound. The pharmaceutical chemical sciences, both phases are usually solvent, most commonly plus solvent, water, non-plus solvent, solvent optical. Partition coefficients are useful in estimating the distribution of drugs within the body. Hydrophobic drugs with high octanol water partition coefficients are mainly distributed to hydrophobic areas such as liquid bilayers of cells. Conversely, hydrophilic drugs, that is the low water or octanol water partition coefficient, are found primarily in aqueous regions such as blood serum. So that can be derived through this experiment. Partition solute octanol and the solute in water. Log P will be log 10 of the solute of the concentration of the solute in octanol and water. This is the ratio. A more polar hydrogen compound will have a lower log P. Value can even be negative and space. More non-polar hydrophilic compound will have a higher log P and will partition into organic groups. Typical values range from 3 to 7. Log P also depends on substituents, hydrophilic or hydrophobicity constant. Pi log P, substitution constant, minus log parent compound. So this is some compound which is having hydrophilic property as values. Now there are some another parameters we are going. Hammett electronic parameter, that is not so much relevant to your topic or your syllabus. So different ratio that you need for the synthesis in classes. Now Tufts steric parameter ES. The Tufts equation is a linear free energy relationship LFER used in physical organic chemistry in the study of reaction and in the development of quantitative structural relationship for organic compounds. It was developed by Robert W. Kraft as a modification to the Hammett equation. While the Hammett equation accounts for how field inductive and resonance effects influence reaction rates, the Tufts equation also describes the steric effects of the substituent. The Tufts equation is written as like RCO CH3H2O, RCOO CH3COH. Here ES is log of partition coefficient of the first molecule with partition coefficient of the liberated molecule. Okay. So this is the graph log K is by log CS3. That is the main complex log K is determined, then specific complex log K is determined. Now, Hans analysis is one of the analysis that is QSL based Hammett relationship. Utilize electronic properties as the descriptor of the structure. Difficulties were encountered when investigator attributed to apply Hammett type relationship to biological system, indicating that the structural descriptors were necessary. In 1962, Hans et al. Entered the scenario with the new and electronic and steady for the model development. This general form of Hans equation is as like this. Log B log P is sigma is CES and constant. Their version coefficient is log P, Hammett constant is log sigma, tough steric parameter is S. Hence model correlate value attributive what is the coefficient ABCD and constant analysis. There are different analytical parameter free Wilson analysis. Then pharmacophore modeling that is been designed through the lead compound. So after this we are completing here. Now we have to study for detailing. Thank you everyone. You can go for the enzymatic study.